Does that work?
Yes, it does. Thank you. All right. So my name is Mikala Capage. I'm an undergraduate researcher in the Garcia lab. And the title of my presentation is Hunting for Prions, Using Inheritance Patterns in Yeast Cells to Attribute Epigenetic States to Prion Proteins. So before I launch into an explanation of my project, I'm going to explain how I thought about framing this presentation. So my research project is an ongoing one. So today I'll be sharing some background information for the presentation, the motivations for this project, and a description of the methods that I'll be using. The central dogma of molecular biology teaches us that information flows from DNA to RNA to protein. And the central dogma really places DNA as the origin of a cell's phenotype, or the characteristics or traits that a cell shows. Recent research has really complicated this picture. We now know of examples in which the phenotype of the cell changes, but there are no corresponding changes in the DNA code. These are examples of epigenetics. Epigenetics is the study of traits that do not arise because of changes in a DNA sequence. And one example of an epigenetic mechanism are prion proteins. Proteins often have two conformations or versions in which they can exist. There's a naive protein, which is the non-prion version, and then there's a prion protein, which is folded differently than the naive protein. Prions occur every so often in a cell, but they have a higher chance of occurring if their naive protein is overexpressed. So if there's more of the naive protein within the cell. And my mentor, Dr. David Garcia, took advantage of this fact in an experiment in which he overexpressed more than 70 proteins, grew the cells for 30 or more generations, exposed these offspring cells to 60 different chemical stressors, measured the growth rate of the cells, and this experiment produced more than 3.5 million data points. And out of all of these results, the Garcia lab has pulled out 5 strains that show a heritable growth trait. So to determine if a strain is showing a growth trait, we perform a growth assay. Jacob Evarts, another undergraduate researcher who's worked on this experiment and these projects, has performed many of these experiments. So to perform a growth assay, you start by placing cells in the wells of a plate filled with a liquid, nutrient-rich media, and then you slide the plate into a plate reader, which sends a beam of light down through the well to measure the optical density of the liquid inside it. And optical density, it is a proxy for cell growth. And the plate reader does this every 2 hours. The data points are graphed, like the graph that I'm sharing on the screen, which was taken from one of Jacob's experiments. And so the orange line is the growth curve for cells that had a prior overexpression of one of their proteins, and the blue line is for cells that had no prior overexpression. And so any differences between these lines is evidence of a growth trait in one of these 5 strains. So the 5 strains that showed heritable growth traits had one of their RNA modifying enzymes overexpressed. And RNA modifying enzymes chemically alter RNA molecules after the RNA is transcribed from the DNA. And this wasn't a coincidence that the 5 strains all had one of their RNA modifying enzymes overexpressed. Dr. Garcia chose these proteins on purpose because the Garcia lab is interested in understanding RNA modifying biology because of the potential that these proteins have to change the expression of many genes at once. So at this point in the project, the Garcia lab has 5 strains that are showing a heritable growth trait. And we suspect that the growth traits are being caused by a prion protein, a prion version of one of the cell's RNA modifying enzymes. But we don't know that for sure. And we'd like to know if we can attribute these growth states to prion proteins for a couple of reasons. One, the results from this experiment have the potential to expand the list of known yeast prions. And 2, all of the putative prions have homologs of their naive proteins in human cells. So understanding how prion proteins can affect a cell's growth in a model organism could one day translate into understanding the role that prions play in human health and disease. So my experiment will be to test these 5 strains to see if they show some characteristics of prion proteins. To conduct these experiments, I'll use the budding yeast, Saccharomyces cerevisiae, as my model organism. So there are a couple of ways to distinguish between traits that are caused by genetic mutations and traits that are caused by prion proteins. And the first difference is that these 2 causes of traits inherit in different patterns. So mutations in the DNA sequence inherit in a pattern of Mendelian inheritance. So if one of the strains in the experiment is wild type, meaning that it doesn't have the mutation, shown in green, and the other strain has the mutation, shown in purple, and these cells are crossed together and then undergo meiosis, the mutant phenotype will inherit in a 2-to-2 ratio. So half of the cells will show the mutant phenotype and have the mutation in their DNA, and the other half will show the wild type phenotype. Now that's really different than how a prion protein inherits. Prion proteins inherit in patterns of non-Mendelian inheritance. So if you do the same type of experiment with a naive strain, so one that doesn't contain prion proteins, and a prion strain that does contain potential prion proteins, you cross these cells together, force them to undergo meiosis, the prion will inherit in a 4-to-0 ratio. So all of the cells will contain the prion and show the prion phenotype. And so performing these experiments is a really good way to test if the strains could be attributed to prion proteins by testing the inheritance pattern of the trait. So that's the first experiment that I'll be doing. I'll be studying the inheritance of these growth traits using yeast meiosis. And to do this, I use a protocol called the Tetrad Sporulation and Dissection protocol. So in this experiment, I start off with a naive strain of yeast that doesn't contain the prion protein, and the experimental strain that has the potential prion proteins marked in yellow. And I cross these 2 haploid strains of yeast together to form a diploid strain. These diploid cells are first grown in a nutrient-rich media and then quickly transferred to a nutrient-poor one. This process is called sporulating and forces the cells to undergo meiosis and produce 4 genetically unique spores encased in an ascus wall. This is called a tetrad. These tetrads are then dissected using a special microscope, and the 4 haploid spores inside of them are broken apart. And then each one of these haploid spores can be put through a growth assay independently to see how many of the spores inherited the growth trait that we saw in the original haploid strain. And so if the growth trait inherits into all of the 4 spores in a 4-to-0 ratio, this will be a piece of evidence that suggests that this growth trait is caused by a prion protein, not a mutation in the DNA. So the next point of distinction between genetic mutations and prion proteins is that genetic mutations are located in the nucleus of the cell, while prion proteins are heritable from the cytoplasmic reservoir, meaning that they exist in the cytoplasm of a cell. And so to test sort of what part of the cell is holding the element of inheritance, you would need some protocol that lets you mix together 2 strains of yeast, but keep their respective nuclei from fusing into one. And luckily, there is an experiment that can let you do this. It's called a cytoduction. So cytoductions are mating without mixing genes. So to perform a cytoduction experiment, I'll start off by taking the experimental haploid strain of yeast, again with those potential prion proteins marked in yellow, and I'll introduce a loss of function mutation into the karyogamy gene. And this mutation restricts this nuclei from fusing with another nuclei. Then I'll take a naive strain of yeast, so one that doesn't contain the prion proteins, and this naive strain of yeast will have a wild type nucleus. So no mutations in this nucleus and no prion proteins in the cell. And these 2 haploid strains of yeast will be crossed together to form a diploid cell that has 2 separate nuclei within the cell. So the nuclei haven't been able to fuse into one because of that loss of function in the karyogamy gene. But the cytoplasm of the cell has been free to mix together. So the next step is to cure off the nucleus containing the loss of function mutation in the karyogamy gene to leave behind a haploid cell that has the nucleus from the wild type strain, but the cytoplasm and anything that was in the cytoplasm from the experimental strain. So then these haploid cells can be put through a growth assay to determine if they show the growth phenotype that the original haploid strain contained. And if they show that same growth trait, then that will be evidence that the trait was being carried in the cytoplasm of the cell, not the nucleus. And that's really characteristic of prion proteins.
Mikala, I just want to let you know that you have 2 minutes left.
Thank you. So in conclusion, these experiments will tell us if the traits that we've observed are prion-based. And if they are prion-based, we'll investigate if the prion form changes the function of these proteins. So to finish up, I'd like to acknowledge the work that I talked about in this project done by Dr. David Garcia and Jacob Everts. I'd also like to thank Dr. Garcia for his unending mentorship of all of his undergraduate students, including myself. I'd like to thank Adam Fries of the Genomics and Cell Characterization Core Facility for helping with a lot of my projects, as well as members of the Garcia Lab for their advice and feedback on this presentation. Finally, I'd like to thank the Europe Mini Grant Program and the Office of the Vice President for Research and Innovation for funding the materials and supplies to complete this project through their mini grant program. And finally, I'd like to thank the Undergraduate Research Symposium team for making this virtual symposium possible. So now I'll open it up to any questions, and thank you very much.
So we will be holding questions until the end, but if anyone has any questions that they would like to ask for us to hold to the end, you're welcome to add them to the chat. Great, thank you so much. Wonderful presentation. And next we have up is Maddie Scott from chemistry.
Thank you. Yeah, let me share my screen really quick here. Okay. Can everyone see that okay? Okay, great. Okay. So yeah, as like was said, my name is Maddie Scott, and today I'm going to tell you about how to quantify the spatial morphology of organic films through polarization-dependent imaging. So that's kind of a mouthful and a lot to understand, so we'll unpack kind of what that means. To give you a brief overview, what I've done is I've developed a technique for characterizing the uniformity of films that we make in the lab using a combination of experimental and computational methods. So the broader application of this work is kind of related to the development of electronic devices. So things like solar cells, organic light-emitting diodes, or flexible electronics, things that are becoming increasingly prevalent in our contemporary society. So one way we can prepare semiconducting materials for these devices is by solution deposition. So solution deposition is a cost-effective and scalable technique for producing films. One technique of solution deposition you can think of as like an inkjet printer, so it's pretty efficient, pretty quick, and would be really ideal to implement on a wide scale. However, what keeps it from being widely implemented is that it often results in severe morphological disorders, so the variety of sizes and shapes and orientations of crystalline structures within the film. So on the right here, we have two example films, a very homogeneous film where we don't see a lot of like macroscopic divisions between domains, and a very heterogeneous film where we can see by eye all of these different grain boundaries and individual structures within the film. So the kind of overarching goal of this project is to characterize morphological disorder in solution deposited films. Further, we want to characterize this so that way we can connect macroscopic changes to the environment, so things like temperature and solution concentration, to the formation of these domains within the film. We're really interested in this because the spatial morphology will dictate the electronic functionality of the film in devices. So if you have a lot of grain boundaries and a lot of structural defects, you're going to severely inhibit the functionality of your solar or your semiconducting film for use in devices. So in order to be able to develop this method, we've chosen a model system called TIPS-penicine. So we've chosen this model system because TIPS-penicine structures, kind of zoomed in here in this image, have polarization-dependent transmission, which means that depending on the orientation of the crystal in space, that will dictate how much light of a particular polarization angle is coming through the sample. To illustrate this a little bit further, we have a schematic of a TIPS-penicine film with discrete aggregate regions shown. And depending on the light polarization and the geometry and orientation of the domain in the film, that'll dictate how much light is coming through. So you see in these little domains here, we have light completely transmitted. And in these domains here, we have light that's completely absorbed and not really transmitted at all. However, if we rotate the polarization by 90 degrees, then we can see the reverse effect. So anything that was completely transmitting before is now completely absorbing and vice versa. So this becomes really useful when we want to connect some sort of measurement strategy like absorption or transmission to the orientation of different domains in space. So by using TIPS-penicine as our model system, we can then develop a joint experimental computational method for developing or for measuring spatial heterogeneity in these films. So it's kind of an overview of all the steps that go into this method. First, we deposit TIPS-penicine films. We then image their transmission using a polarization-dependent brightfield microscope. We then fit all of this transmission data to some sort of sinusoid. And from those fitting parameters, we can then identify distinct aggregate domains within each sample to characterize then the spatial morphology. So this kind of first step here in which we deposit the TIPS-penicine films. Our deposition procedures are pretty simple. We just clean the slides with methanol. We then put on some sort of wetting layer called PETS that helps the TIPS-penicine solution stick better to the surface of the film. And then we drop cast our TIPS-penicine solution at specified temperature and concentration conditions, let the solvent evaporate off, and then we form this nice sort of crystalline structure here. Once we've developed all of our films for analysis, what we do is we image them using a polarization-dependent brightfield microscope. So there's a lot going on in this image, and I won't talk through all of this right now, but if you have questions after, happy to answer those. The basic sort of gist of the microscope is that we have light coming in from a light source. We filter out one polarization using a linear polarizer on a rotation mount. We then have the light come through the sample and record its transmission with a camera. So we actually don't have any magnification in this microscope because we want to be able to image as much of the sample in one shot as possible, and then image several spatial locations along the sample to be able to stitch together and get a comprehensive composite image of the whole film. And I'll talk a little bit about that in a second. So with this microscope, what we do is we collect 18 different polarization-dependent images at one spatial location. So this little GIF here is showing at one spatial location on the film, we have 18 different images of the transmission. And already from this image, you can start to see different features light up and darken together, indicative that there are discrete orientations of domains within the film. So what we can do is, since transmission is periodic, we can fit this data to a sinusoidal curve. And we fit each individual pixel, the transmission, as a function of polarization angle at every pixel in our image to some sort of sinusoid shown here on the left. By doing that, what we can do is generate fitting parameters, including the amplitude, phase shift, and vertical offset as a function of space in that image there. So kind of shown here on the right, we have a spatial image of all the different phase shift parameters for that single kind of GIF that I showed in the previous slide. So once we've done that, what we can do is, stitch together several spatial locations to kind of collect a retroactive panorama shot of our film. So this enables us to look at the entire film all together and characterize domains over a larger region of space. So using a composite map for domain identification enables us to better observe how macroscopic perturbations, so things like solution concentration and deposition temperature, affect the mesoscopic structural organization, so the different domains within our films. So once we've taken these retroactive panorama shots, what we want to do is be able to pick out all of these different features, which we can kind of see by eye. However, kind of sitting down and picking each one out by hand is going to take a really long time. So what we've done is we've automated this process using a flood fill algorithm. So essentially what a flood fill algorithm does is it takes a single pixel and fills out an entire shape. So if you've ever used like Microsoft Paint, that little paint bucket tool is kind of the same idea. So we kind of start with a single seed and compare it to its neighbors and set some sort of threshold for comparison. So in our case, it's usually about 3 to 5 degrees of similarity between pixels. So we'll look at this entire spatial map of phase shifts. And for that map, we'll fill out the different domain assignments based upon how similar each neighbor is to a seed. So as we can see here, our flood fill algorithm does pretty well and picks out those major domains that we can start to see by eye. And the beautiful thing is we don't actually have to press any buttons and do anything ourselves. So from this, we can start to characterize spatial disorder by looking at looking at some sort of metrics for the spatial domains that we see in the film. So in order to do that, what we've done is we've quantified the size, the aspect ratio, and intra-domain phase gradation of these different domains within the film. So the size being sort of just the total number of pixels, add them all together, and give them some sort of physically meaningful characteristic like millimeters squared. For the aspect ratio, what we're looking at is sort of the ratio of a length to a width of a domain.
Okay.
And essentially what we want to do is use some math to be able to do that independent of orientation. And if you're interested in that, I can talk a little bit about that later too. Kind of our third metric for characterization is intra-domain phase gradation. So as you can see here, the single domain has a lot of variation in the phase assignments. So what we want to be able to do is kind of map that out and see if there are any connections with the macroscopic perturbations. So unfortunately, the data I collected doesn't really show a lot of these trends very well, but eventually what we want to do is connect these mesoscopic spatial kind of disorder to macroscopic changes, kind of like what I've been saying. So by eye, by comparing these different films, we can see that there's a change with regard to temperature. However, we want to be able to use these different characterization metrics to be able to quantify that and make it more reproducible. So in summary, what we've done is we have used a polarization-dependent microscope to collect images of the transmission of a TIPS penicillin sample.
Maddie, just so you know, you have 2 minutes left.
Thank you. We have used a home-built image processing and analysis package to use this information of the phase shifts as a function of space to separate pixels into discrete domains. Then what we do is we characterize the size, aspect ratio, and inter-domain phase gradation as a function of macroscopic parameters, so solution, concentration, and temperature. And ultimately, what we want to be able to do is connect those things such that we can further quantify the relationship between environment and mesoscopic structure, and increase the reproducibility of solution deposition techniques such that we can further implement them in the production of electronic devices. So with that, I'd like to thank members of the Wong Lab, specifically Dr. Kathy Wong and my graduate student mentor, Kelly Wilson, who's here today, for their exceptional guidance and mentorship in this project and my time in the lab. I'd also like to acknowledge the funding sources at the University of Oregon, as well as external funding that I've received to make this project possible. And with that, I'd like to thank you all for watching.
Thank you very much for that presentation. If anyone has questions, you are welcome to add them to the chat, and we will make sure that they are asked at the end during our Q&A. So we have Nora up next. Nora, the mic is yours.
Okay, great. Let me share my screen with you. Are you seeing my screen? Okay, we'll assume yes. Hi, my name is Nora Kearns, and I'll be presenting my research on the influence of prenatal inflammation on postnatal maternal and infant behavior. With the rise in obesity and overweight, an increasingly common risk factor during pregnancy is maternal BMI. Today, over half of American women begin their pregnancies either overweight or obese, and as this graphic is showing here, these frequencies are steadily rising. And while the immediate risks that obesity poses on the health of mother and child are recognized, risks like gestational hypertension, diabetes, and labor complications, less is known about the lasting implications of maternal metabolic state and the in utero environment characteristic of obesity on maternal and child long-term health outcomes. Obesity is characterized by chronic systemic low-grade inflammation. Such inflammation is increasingly recognized as a contributor to psychological illnesses like anxiety and depression. And inflammation provides a mechanism that can link obesity to mental health. So the questions that we're interested in exploring are how maternal gestational inflammation, which can be characterized by levels of cytokines and fatty acids, influence postnatal mother and infant behavior. A major reason which drives our need to understand the influence of chronic systemic low-grade inflammation on mother and child mental health is our knowledge of maternal immune activation. Acute, severe inflammation in the mother, which would occur in response to a pathogen, has been linked to autism spectrum disorder and schizophrenia. These immune challenges, which are characterized by elevated levels of pro-inflammatory cytokines, can exert detrimental effects at varying time points. We know that the fetus in early development is incredibly sensitive to environmental stimuli, so inflammation in the mother can affect development of neural circuitry and synaptic plasticity in the fetus at critical time points, like during neurogenesis and synaptogenesis. However, we know a lot less about chronic inflammation and its effects during development. Adipose tissue secretes cytokines at a level proportionate to fat mass, and the composition of cytokines changes as BMI increases, such that obese individuals tend to secrete more inflammatory cytokines like interleukin-6 and tumor necrosis factor alpha. Adipose tissue also attracts macrophages to dead adipocytes, which accumulate around the adipocyte in crown-like structures and then secrete interleukin-6, tumor necrosis factor alpha, and other inflammatory cytokines. And of particular interest to us is the fact that many of these inflammatory cytokines play a role in regulating neurotransmitters involved in the regulation of mood, including the 5-HT system. So pro-inflammatory cytokines like IL-6 and TNF-alpha can reduce survival of 5-HT neurons. They activate a metabolic pathway which has the same precursor, tryptophan, as the serotonin synthesis pathway. So activation of the kynurenine pathway effectively steals tryptophan, which would have otherwise been destined for serotonin synthesis. This results in reduced serotonin neurotransmission and subsequent alterations in mood. Reduced 5-HT is associated with anxiety, depression, autism, and ADHD. However, it's believed that polyunsaturated fatty acids like omega-3 fatty acids may play a protective role. Omega-3s are recognized for their ability to reduce inflammation and decrease the levels of inflammatory cytokines secreted by immune cells. Rodent studies have shown that mice who experienced maternal immune activation, who were then fed a PUFA-enriched diet, experienced none of the consequences of the maternal immune activation. Those who were fed a standard diet displayed elevated anxiety. Rodent research also shows that omega-3 adequate mother mice exhibit more nurturing behaviors towards their pups than their omega-3 deficient counterparts, who are actually more likely to attack or fail to nurse their young. So we want to see if this anti-inflammatory effect can translate to more positive maternal behavior and child development. The data from my current research comes from a pilot study conducted by Eleanor Sullivan's team at OHSU. 62 women in the study were recruited, 33 of whom had ADHD, which allowed us to also study relationships between inflammation and ADHD in mother and child. Maternal pre-pregnancy BMI was obtained from medical records, and maternal inflammatory state was assessed using plasma fatty acid and cytokine levels from second and third trimester blood draws. We assessed levels of 3 inflammatory cytokines, which are shown in orange, as well as 1 anti-inflammatory cytokine, interleukin-10. We also assessed many different polyunsaturated fatty acids, but I will broadly categorize these into omega-6, which are categorized as inflammatory, and omega-3s, which are anti-inflammatory. To assess maternal and infant behavior, a variety of tasks were performed at postnatal time points. I focused on the mother-child free play, in which the mother was asked to join her infant on the floor for 10 minutes of play as she would given free time in her day. And this was done to kind of evaluate how the pair would normally behave. Using a well-validated coding scheme, I assessed mothers based on characteristics like sensitivity and intrusiveness, with scoring from 1 to 5. And similarly, scoring from 1 to 5, I assessed infants on characteristics like positive and negative mood and sustained attention. These were all conferenced with at least 1 additional scorer, and maternal codes were evaluated with 2 additional scorers. We observed expected correlations between inflammatory cytokines and BMI, seeing that mothers with higher BMIs generally had high levels of all 3 cytokines, and these are correlation coefficients here. A latent variable representing total inflammatory cytokine profile in the 2nd and 3rd trimester demonstrates that BMI was consistently and strongly correlated with inflammatory cytokines. Mothers with higher BMIs generally demonstrated lower levels of omega-3s, EPA is one kind of omega-3, and a lower O3 to 6 ratio. Higher omega-6 ratios higher omega-6 levels and omega-6 to 3 ratios were also observed in mothers with higher BMIs, and mothers with higher BMIs were less likely to supplement with an omega-3 fatty acid. So generally, we're seeing expected correlations between inflammation and BMI. We observed many correlations between omega-3s and maternal BMI, so there's kind of a lot to unpack here, but I will turn our attention to what I feel are the most significant and interesting findings. Consistently, we observed that omega-3s were correlated with positive maternal behaviors like sensitivity, positive regard, and animation. Composite sensitivity is a latent variable which kind of represents overall positive parenting by combining these 4 positive parenting scores and subtracting for the negative ones. Also, what I feel is important is that fatty acid supplementation was correlated with sensitivity and negatively correlated with intrusiveness and negative regard. We did not observe many correlations between cytokines and maternal behavior, though interleukin-10, which is an anti-inflammatory cytokine, was positively correlated with sensitivity. The beneficial effects of omega-3 fatty acids, which were observed in maternal behavior, generally did not translate to modifications in child behavior during free play. However, we see some interesting results regarding infant activity with maternal BMI, ADHD, and tumor necrosis factor alpha. Though not directly related to infant and child mental health, there are a couple of interesting relationships here. So mothers with ADHD who typically demonstrate higher BMIs and higher levels of TNF alpha, we see that their children demonstrate reduced physical activity at this very early age. There is a heritable component to ADHD, and while one might expect that a child with ADHD would be more physically active, they are actually at a higher risk of developing childhood obesity, and exposure to low-grade systemic inflammation during metabolic programming in utero may explain this correlation. Collectively, these results suggest that omega-3 fatty acids and supplementation during pregnancy could attenuate the potential effects of inflammation and contribute to more optimal maternal mental health and behavior in the postpartum period. Additionally, fatty acid supplementation could be a really simple and effective dietary intervention for mitigating the influence of inflammation on maternal mood. A more thorough investigation is required to draw conclusions about the effects of omega-3s on the infant, but the findings observed on maternal ADHD and TNF alpha suggest that inflammation could contribute to child physical lethargy, which could be a mechanism that explains how maternal obesity predetermines childhood obesity risk and the heightened obesity risk for children with ADHD. One limitation of using the free play interaction is that it's not really designed to elicit a broad range of emotional regulation from the child, because typically playing with their mom for 10 minutes is, or hopefully, a pretty normal activity for them. So in the future, I'd like to look at other behavioral assessments that do allow the child to demonstrate a greater range of emotional behaviors.
Nora, just so you know, you have 2 minutes.
Okay, thank you.
You're welcome.
This research was done on a pilot study for Dr. Eleanor Sullivan and Joel Nigg's PEACH study, which I was involved with this past summer at OHSU. The PEACH study will obtain data from approximately 300 mothers and will have more robust biological and behavioral data that could allow me to study infant behavior in a greater range of tasks designed to elicit a broader spectrum of emotional regulation. I'm also very interested in epigenetics and inflammation and its role in the regulation of gene expression. So as the PEACH team collects placentas and umbilical cord samples, I would love to study maternal diet and infant epigenetic modifications. I would like to thank the University of Oregon Office of the Vice President for Research Innovation for sponsoring my research project. I would also sincerely like to thank Dr. Sullivan and Dr. Gustafson, who have inspired my interest and desire to pursue research and my passion for maternal fetal medicine, and for all the support they've given me over the past 2 years. Thank you so much.
Thank you, Nora. Awesome presentation. Alrighty, we have one final presenter. And before I make their introduction, if you have any questions for Nora, please add them to the chat. We'll make sure that they get answered in our forthcoming FAQ. All right, and our next presenter is Isabelle, Isabelle Cullen. All right, so the mic is now yours.
All right. Can you guys see my screen? I think so.
You're good.
Okay, awesome. Hi, my name is Isabelle Cullen. I'm a student in the SMEAR lab in the Institute of Neuroscience, and today I'm going to talk to you about my research project called Modulation in Sniffing Behavior as a Biomarker for Autism Spectrum Disorder. Shoot, come on, presentation. There we go. So autism spectrum disorder is a neurodevelopmental condition characterized by a variety of symptoms. So for example, my brother Davis actually has autism. And for him, his autism shows up as hyperfixation on things such as English culture and artwork. This is actually an example of art he made himself. Social deficits such as being unable to hold the natural back and forth of a conversation or maintaining eye contact during a conversation, and sometimes repetitive motions. He tends to clap his hands a lot when he's excited. And autism is currently one of the most common neurodevelopmental disorders affecting 1 in 54 individuals. So one of the challenges to diagnosing autism spectrum disorders is these children are generally diagnosed between the ages of 2 to 4 years old. And you would think you'd be able to know even earlier. However, these children do present as neurotypical at birth, and they start developing the onset of symptoms around 12 to 18 months. So we see this later onset and also later diagnosing period. So why we're trying to find a biomarker, like I said, is to assist with earlier intervention. So biomarkers are a broad subcategory of medical science that allow us to accurately detect or even earlier detect a condition. So for example, the quantity of chloride in sweat is actually used to diagnose cystic fibrosis, and currently we do not have one to diagnose autism spectrum disorder. So why we think olfaction or your sense of smell would actually be a wonderful one is it's one of our most primitive senses and highly conserved across animal groups, even including bacteria. In autism, olfaction has actually been used as a biomarker before for schizophrenia, Alzheimer's disease, and Parkinson's. So one of the previous pieces of research that has primarily informed our research is Rosenkrantz 2015. So Rosenkrantz, along with other research, has found that when you smell attractive things such as roses, soap, or cookies, you actually increase your sniffing magnitude, so you take in more air. Whereas when you smell aversive things such as trash or rotten fruit, you actually decrease your sniffing magnitude, so you take in less air. And the goal of this paper was to investigate, do children on the spectrum smell and perform similar sniffing modulations to neurotypical kids in accordance to previous research. So how they measured this was using a olfactometer. So they connected the children to the olfactometer and had them watch cartoons. And they were connected to a small intranasal cannula device, which both simultaneously delivered the odorants and also recorded their sniffing behavior. So they would trigger the odorant itself at the onset of inhalation to measure the magnitude of the sniff following, wait 30 seconds, and then trigger the next odorant. And they did this for about 10 minutes with a group of neurotypical children and children with autism. So what they found was typically developing children, or neurotypical children, as I've been referring to them to in figure A, they did respond similarly to previous research in that they had an increased sniffing volume to pleasant odors and a decreased sniffing volume to unpleasant odors. However, they found something different for the children on the spectrum. They found that children on the spectrum did modulate their sniffing behavior to attractive odorants. However, they did not to aversive odorants. So this led to the question, was this a difference in perception of the odors, or is this just an altered motor mechanism? So they tested this by asking these children to rate each odorant on a pleasantness scale, and they could tell you that this was a pleasant or unpleasant odor. However, they still performed a different sniffing modulation. So where our research and this meiosis project comes in is we're trying to understand the neural mechanisms behind this phenomenon observed in the Rosenkrantz paper. And how we plan to do that is using a mouse model rather than a human model, because it's a little bit difficult to go poking and prodding into people's noses. But using a mouse model allows us to have more control over the neural system and also use more tools in order to look at the neural system of the olfactory bulb. And plus, olfaction is actually the primary sense for mice, so this makes it the perfect model organism to use for our research. So like I said, we're using a common B6 mouse, essentially our wild type mouse, as our model for neurotypical children, and trying to establish a baseline to see, do these mice actually perform similar sniffing modulations to the neurotypical kids? So how we were planning to do this is using a small device called a thermistor. So a thermistor is a small device that allows us to track changes in temperature. So inhalations tend to be cooler, exhalations tend to be warmer. So through that, we can indirectly measure the modulation in sniffing behavior. And every mouse will also be implanted with a head bar, which essentially helps keep them still during the actual paradigm. So for the experimental setup, once the mouse is recovered from surgery, they will then be placed in our experimental box and placed in a small little device that we use to keep them head fixed, so they'll be still during the paradigm. Then they'll be scooted up to a device where the odorants will be dispersed at them, and because the Rosenkrantz paper looked at passive sniffing behavior, we will also be looking at passive and naturalistic responses to these odorants. So that's nice because we actually don't have to train the mice to do anything. They just have to be in the box and be exposed to these odorants. So the odorants that we will use are previously categorized odorants as aversive or attractive to mice. So for our unpleasant odor, we will use 4-methiazole, which is a sulfur compound. The mice tend to avoid this when they're in presence of it. For our attractive compound, we will use 2-phenylethanol, which is a component of roses. And for our control, we will use clean air. So like I said, in the Rosenkrantz paper, they studied both neurotypical children and children with autism. Once we've established the baseline using the B6 mouse, we will then use the Fragile X knockout mouse to be our model for autism. So this is a great model for autism, especially because Fragile X syndrome is an intellectual disability caused by mutation on the X chromosome. And currently, males with Fragile X have a high comorbidity rate with autism spectrum disorders, anywhere from 30 to 54%, depending on the paper. And this makes it the largest single gene contributor that we know of today to autism spectrum disorders, making it the ideal model to use for our project. In addition to this project, we will also be tracking orofacial movements. Orofacial movements meaning, jaw and facial features themselves. Previous research has denoted that there's a link between orofacial movements and breathing patterns, and we want to see if the same is true for olfaction. So as you can see in the figure on the left, that mice actually do have different facial features in response to certain things, such as different tastes, different stimuli, such as a tail shock. in different odorants possibly. So we want to see if this is true for olfaction. So how we plan to do this is using a small device called a Raspberry Pi camera. A Raspberry Pi is essentially a small operating system that allows the user to do whatever you want with it. So for us, we will be coding to make it into a camera to record orofacial movements during the same paradigm that I described previously. So this is an example of the software that we will use to actually track the orofacial movements in the mouse. So this is called Deep Lab Cut, and it allows you to pinpoint specific parts of the mouse or object that you really want to track. So this is an example taken from our lab from a previous paper. So you can see it's pointed at the nose, the center of the head, and the center of body mass. It can actually track the track the body position in real time. So we hope to use this for orofacial movements, and we'll specifically track the nose, the jaw, vibrissae, ear position, and the eyes, and seeing how these change in response to attractive and aversive odorants. So in summary, we're looking at the neural mechanisms of the biomarker described in the Rosenkrantz paper for autism spectrum disorders. And the Rosenkrantz paper found that children on the spectrum do not modulate their sniffing behavior in response to aversive odorants. So with our research, we are trying to understand these neural mechanisms, and how we plan to do that is using a common B6 mouse as a model for our neurotypical children, and then repeat the same paradigm using a Fragile X knockout for our model for autism. And we're essentially looking at the changes in olfactory patterns in response to attractive and aversive odorants. And we will also look at orofacial movements in response to these attractive and aversive odorants using a Raspberry Pi camera. With that, I want to acknowledge a couple of people. I'd really like to acknowledge the Peter O'Day Fellowship and UROP for funding my research for this upcoming summer, but also the Smear Lab, so Dr. Matt Smear, for all of his mentorship and help in answering my endless questions about everything, but also Jared Acosta King, who is my graduate student and helping me with this project, who's been instrumental in creating and designing this project, but also the Robert D. Clark Honors College and the Department of Biology for allowing me to pursue this as my thesis. And those is my presentation.
Awesome. Thanks so much, Isabelle. That was, like I said, all the presentations were fascinating, but that was a really great presentation. Thank you so much. So now we are, I know we started out a little late, but we are going to have time for that Q&A. So I'd ask for all the presenters to please turn your videos on, and also your mics too, for our presenters only. So I have some questions that I would like to ask all you researchers, but I'm curious from the audience if anyone has a question. And it looks like there is one in the chat from Maddie Scott. And so Maddie, would you like to ask your question for Isabelle?
Yeah. So I was just curious, kind of like what the uncertainty in position is when you're tracking the orofacial movement in the mice, like, and how are you able to resolve the different, like you had mentioned, different parts of the mice, like as you're tracking it?
Yeah, so that's an excellent question. The plan was to start looking at that this term. So we haven't actually had the chance to do that yet. However, we're actually going to work with the McCormick lab this term, for the rest of this term and during the summer, to start creating the deep lab cut data to see how, what it does look like, because vibrissae or whiskers are essentially very thin and small. So it is a bit of an issue trying to track those. Previous research has also painted them. So we're going to see if that's maybe a possibility that we can use for whiskers and tracking that.
Awesome, thank you. And again, if anyone has questions for our presenters, you're welcome to add them to the chat. So this is a question for all four of you. So feel free to jump in, any of you. So here's some questions. So could you tell us about how you got interested or involved in your research? And I guess let's start with Mikala.
Sure, that's a really good question. I am a sophomore this year, and since I started at the U of O, I knew that I wanted to pursue research here at the university. I was really interested in what research could teach me about science, kind of beyond the classroom. And so last winter quarter, I got involved with the Garcia lab. And the Garcia lab is very new. And so this lab really gave me the opportunity to spearhead my own project under the mentorship of Dr. David Garcia. And I really enjoy doing research at the university and hope to continue in the next couple of years for sure.
Cool, that's awesome. How about you, Maddie?
Yeah, so I got into research actually kind of by accident. Like I was taking general chemistry and had come into the university like 4 years ago thinking I wanted to be a medical doctor. Then just like fell in love with like electrons and kind of thinking about like electron shells and what that even was. And so I ended up kind of finding Dr. Kathy Wong's lab, who specializes in kind of using lasers to understand the electronic properties of different materials, and just absolutely loved it and have been working in her lab for like the last 4 years. And so yeah, that's kind of how I got involved in what I'm really interested in.
That's rad. That's awesome. How about you, Nora? Tell us about how you got interested or involved in research.
Yeah, so I was in a class with Dr. Sullivan my freshman year, a nutrition class, and I've always been really interested in nutrition and in women's health, and her research is kind of the perfect combination of that. And so I started with her at the end of my freshman year, and then after my sophomore year, I got to spend a summer working with her team up at OHSU, which was such a wonderful experience, and such a wonderful community of female scientists too. So I really owe that to her, my interest in research and passion for that.
Awesome. Yeah. How about you, Isabelle?
So mine's kind of, I've been involved in research since my freshman year, but I've actually jumped departments a lot. I've gone from the psychology department to education to now neuroscience. And it's been, it's been a wild ride, but I'm really fortunate to be in the lab that I'm in. I actually was in Dr. Smear's class sophomore year, and I asked him to take a look at one of my documents for internships that I was applying for. And he asked me, would you be interested in my lab? And I said, absolutely. And we ended up talking and having a wonderful conversation about autism. And I told him we had a similar connection to autism spectrum disorders. And that's just kind of solidified my love for my lab and the research that I've done.
Great. So again, if anyone has any questions for our presenters, feel free to add them to the chat. But I've got a few more before we wrap up in the next 10 to 15 minutes. So we've got a question in the chat from John. Outstanding job to all the presenters. Nora, omega-3 supplementation shows benefits in your study. Is there any downside to it? Is the cost reasonable to provide it broadly to at-risk families?
Yeah, that's a great question. So omega-3s I would say there's really no downside, and there are so many benefits too, whether that's mental health or cardiovascular health. And the American diet is really deficient in omega-3s. So I think that's something that everybody should be aware of in their diet. And I think it actually is a really reasonable and cost-effective way, because essentially you can just take a fish oil supplement, and omega-3s are found in so many different foods like walnuts and greens, and plant-based foods. So I think it is a cost-effective or a really inexpensive thing to incorporate into your diet.
Thank you. Good question. Hey, that happens to me all the time, Kayla. I got a dog.
Sorry about that.
No, it's all good. So this is actually not one of my stock questions, but I said in the beginning in the introduction that I was super pumped to be moderating a panel of lady scientists. And so I'm curious if any of you would share, what does being a lady scientist mean to you?
Yeah, sure, I can start if that's okay.
Yeah.
So being a female scientist, especially in physics, is a little weird, at least for me. I've definitely taken classes where I've been the only woman in the class, and it's not something that I really notice until I'm put in that situation where all the guys are super nice, and it's never been like a hostile environment, but it's just something I notice because I become sort of a representation of what it means to be a female scientist in a class. But I think that's actually been very motivating for me because a lot of my friends are also guys. And so it's been a nice push to like, you know, stay on pace and still do really cool things and just make it not something that is a barrier, but more just kind of part of who I am. And I think like, I'm really excited to continue that role, especially I'll be attending graduate school at MIT in the fall. And so I think like, that's a really cool, like, thing that something I guess I was just also aware of going in there because the ratio of men to women is a lot higher, I guess, like than other places. And so it's just been something that I've just become really proud of and not really like something that's become a barrier for me. Yeah.
That's so cool. Congratulations. What an awesome accomplishment for getting into graduate school.
Thank you.
Super, super rad. That's super awesome. I can totally relate to what you were saying. I even though people assume because I'm a librarian, I mostly work with books, but I'm heavily involved in information technology and computer science. And I definitely can relate to everything that you just said. How about any of our other presenters, if you'd like to share?
Yeah, so one of my experiences at U of O is that I've really noticed that in academics and in the classroom, women have a really strong role in teaching, and there's a lot of women in the science classes that I've been taking. And that's been really great to see at the university. But I've noticed that that strong female representation doesn't always carry into research. A lot of the mentors or postdocs or graduate students are male, and so there's this kind of difference between what you see in the classroom with students that are really eager to enter the field and then people who have made it in the field and are mentors. And so it's really great to see a bunch of female students presenting at the research symposium and in this group. And I just look forward to seeing those new generations of students and people who are interested in learning the field, really see them be mentors to the next group of people coming up.
Nora or Isabelle?
Yeah, so I think my research is kind of unique in that I'm like surrounded by women all the time, which is great. Like in the Peach lab, it's pretty much all women, which was such a fun experience for me to see. Just I feel like I've been privileged to have such incredible professional role models, the research assistants on the Peach team, as well as Dr. Sullivan and Dr. Gustafson. And I think it's just always been, I guess I don't think so much about like myself compared to male researchers because I'm not often surrounded by them. But yeah, I'm just constantly surrounded by positive female scientist role models. So that's been a cool experience for me.
How about you, Isabelle?
For me, it's been kind of a unique position being in the Institute of Neuroscience. I'm really fortunate that my lab has really great representation of female scientists. One of our graduate students, now postdoc, just finished her PhD, and she's been a wonderful advisor and mentor to me, especially in trying to code. The camera has been, trying to code in Python is not always the easiest feat. So she's been wonderfully helpful in that. But it's definitely kind of inspired me to continue on in the path of academia because we currently, in the Institute of Neuroscience, we have currently, I believe, one female and like as little females in the actual department. So I'm hoping that, that actually continues to increase as, the future comes on.
Thank you. Yeah, thank you all for answering my question. All right, do we have anyone else that would like to ask our panelists questions? Please feel free to add it to the chat. So I've got another question for y'all. What are your goals and aspirations for each of your research projects? And so that's my question. What are your goals and aspirations?
Well, I guess I'll start. I'm really interested in epigenetics, which is kind of how the genome can actually change over time given regulation by dietary modifications and environmental factors. And I'm really interested in how what mom is consuming during pregnancy can alter genetic expression in the child. So in the future, whether that's right now we're not really sure what's going to happen with research. But sometime in the future, I would really like to focus on that, looking at epigenetic regulation of the fetal genome given the maternal diet.
I can go next. So for my research, we're hoping to essentially understand like the neural basis of this biomarker. And it has been reproduced in humans, but it's been with some mixed results. So we're hoping that going forward, we're able to maybe apply this to humans and see, maybe does this biomarker actually exhibit even earlier before the onset of symptoms and help intervene earlier for children with autism.
One of the goals of my research project and the work of my lab is that prion proteins have kind of a bad reputation. They're associated mainly in people's minds with diseases like Kuru or mad cow disease. But the Garcia lab is really interested in finding instances when prions are actually beneficial to the cell. So my project is setting out to identify some new prion proteins, and we're hoping that we can get a better understanding of how prions work in the cell, both really to look for places where they can benefit how cells grow and change in response to their environment.
Yeah, so one of the big kind of broader kind of goals of my project is really far down the line, being able to sort of control macroscopic changes in a film deposition environment, so things like temperature or solution concentration, and have a more fine-tuned control over the types of organic thin films that you can produce for use in electronic devices, specifically because solution deposition is a really cheap and kind of ecologically sustainable technique for making semiconducting films. It'd be really great to transition to that in a broader scale. And so having a more understanding of how we can control the types of films that are produced will help it to become better implemented in the future.
All right, I got one last question before we all say goodbye and have our wrap up. So my final question is, what is next for you in your research and in your life after the symposium?
Okay. So for me, obviously it's getting back in the lab to, start data collection. So I know we're all hoping to get back onto campus and, restart our data collection, just being in the research labs again. That would probably be the biggest thing. But for me, that would also be working on my honors college and departmental thesis with this project, but also learning how to take the future steps towards graduate school.
So next up, I'm going to finish out the term and write the paper that I've been putting off while working on this presentation. That's what's next. But after that, I hope to be back in the lab this summer under a VPRI fellowship from the Office of Vice President for Research and Innovation to really conduct these experiments that I lined out today, collect the data, and then see what the next steps are for the Garcia lab and for my work.
How about you, Nora?
Yeah, so right now I am a research assistant with the branch of the PEACH study that's operating in Eugene, which is the PPOD study. So we are kind of transitioning our data collection over to remote. So we've been working on that right now. And then next year I'll be working on my thesis through Dr. Sullivan. And then I'm also involved in a bioengineering research project, which I will hopefully be conducting over the summer, fingers crossed. So yeah, that's kind of what's next for me research-wise.
Cool. And how about you, Maddie?
Yeah, so kind of on the short term, I'm defending my honors college thesis next Wednesday, so that's really exciting. And then hoping to get back into lab pretty soon to collect some more data so we can hopefully write a paper about all of this technique and how it works and how it can be used to quantify spatial morphology. Kind of the longer term, like I said, I'll be starting graduate school, hopefully in the fall at MIT, kind of doing similar research to what I'm doing here, focusing on sort of developing different ultra-fast laser techniques, which I didn't really talk a whole lot about that in my presentation, but I love lasers. That's kind of what I want to spend the rest of my scientific career doing, is shooting lasers at molecules and seeing how they respond. So.
Oh yeah, I just had this image in my head of like laser cat, that meme.
Basically want to be laser cat.
Awesome. Well, we are at the end of our session, and I wanted to thank Isabelle, Mikala, Nora, and Maddie for gifting us with your knowledge. And this was like, it's been an honor to be your moderator for this session. And I wish all of you the best of luck with your research and continue to stay safe. Thank you everyone for joining us today. Let's give our colleagues, you can turn your videos on now if you want. Turn your videos on and let's give everyone like a big thumbs up. Good job, y'all. Good job. Yeah. Cool. Alrighty, well, this recording will be made available. So that is the end. Hope everyone has a wonderful afternoon. And thank you for attending the session. And we hope you have, a good time at the other ones. So adios.
Thank you so much.
Thank you.
Great job, everyone.
Bye.